As an introduction, we like to acknowledge that our mission at Vose Library is to advance learning, inspire curiosity, enrich lives, and promote community. With that in mind, let me introduce our guest. With her husband and family, Wendy Reinemann owns and has operated Guini Ridge Farm since 2001. It is a diversified operation that includes raising livestock, market gardens, and the most recent addition, a garden center in Rockport. In addition, Guini Ridge Farm offers a variety of classes and events throughout the year, including their annual lamb debut and Christmas market. Prior to joining the farm full-time in 2017, Wendy worked in both the biotech world, where she got to use her molecular biology degree, and the seed industry, as the Vice President of Product Development for Johnny Selected Seeds for nearly 10 years. Her passion for plants, business experience, and scientific background have proven to be the right combination to continue to grow Guini Ridge Farm. Additionally, Wendy is also the proud parent of 2 grown children and excited that her son is now also working full-time at the farm. So without further ado, let me introduce you to Wendy. Welcome, Wendy.
Hey, how are you? Glad to be here tonight.
Thank you so much for being here.
Excellent. So I'm going to try to get this on screen share.
Perfect.
Okay. So can everybody hear me good? We're all right? Excellent. So I don't know about you, but I get really excited every year when the new plant catalogs come out, the new seed catalogs come out, and you get to see what's new. New colors, new varieties, new strains. It's always kind of fun to see what's going on in the industry and see kind of what breeding work has been happening over that time frame. One of the key things we look at it for our garden center and for our farm is those new varieties that are going to help us in some way, shape, or form, whether that's through yields, higher yields on a product, or a disease resistance package that's going to make that plant a little bit happier and healthier, where we don't need to spray or do any, integrated pest management or anything, other things to take care of that plant. So the question that I get asked a lot of times is, how do they make these new plant varieties? How do they come to be? So this little chat we're going to have tonight is going to hopefully kind of cover some of the basics of plant breeding. Plant breeding in itself is a huge science, and there's a lot of details to it and a lot of things, but this hopefully will give you kind of a good overview of how new plant varieties come to be. So what we're going to kind of cover tonight is the dawning of agriculture and how it really all started with plant selection. This really goes back, to the beginning of time when humans started to actually settle into cities and to villages and towns. Then we're going to kind of talk about how plant breeding moved a little bit forward, where we were looking at modifying desired traits in a little bit more of an organized way. Then we're going to talk very briefly about how it got really complicated and very complex, and some of the things that they're using today to hopefully move plant breeding forward a little bit more quickly. But what you're going to kind of take away from this is it takes lots and lots and lots of time to actually get those varieties bred and into your hands. But at the same time, this is something you can actually do in your own backyard. You don't need to have a big research station to do this. It's something you can actually, enjoy in your own gardens. So really, how did plant breeding all begin? It began, like we were talking about, kind of at the dawn of the Neolithic revolution, when people started forming in villages. And there's been kind of a debate within the industry is what started first and why did this all happen? There's been some science that suggested that the thirst for beer was actually what probably founded plant breeding and formed groups to try and improve the grains and things that are needed to make beer. And then that followed with bread and other things that came down the path. So early humans' thirst for beer kind of started this whole process out, to hopefully gain the right crops and the right quantities so that they could actually create beer better. So what those first farmers did was they went and, in the wild where they were wild harvesting these plants, and they domesticated them. They saved seeds from those plants that had the traits that they were looking for, and then grew those then into the next season. Over time, you can definitely modify a plant to have those traits that you're looking for, whether it's something that might be taller, higher yielding, a specific color. So those accumulate over time, and those new traits, those desired characteristics, are what creates that new variety. Let me know if I'm going too fast. So how did different crops originate? There's something called land races, and those land races are species from the wild populations. And from this graph, you can kind of see where some of the crops that we know today, where they really originated. So for example, potatoes that are now worldwide, and a lot of people think of Ireland, potato famine is where potatoes were, but they were originally found in the Andes Mountains. And were brought back with the conquistadors during the Columbian Exchange, where then they were actually used, developed into more varieties that were more applicable to farming, and then became widely used in Europe. So this is a really great way to kind of have an overview of where these land races and where these wild populations come from. And this is where plant breeders actually go to get some of that genetic material from some of these old wild types that you find in nature. If anybody's been to our garden center, I'm a little bit of a begonia fanatic. Begonias were originally developed in South America and kind of northern Central America. And if you look at the varieties that are there in the wild, you can hardly even identify them as what you would see in a begonia in a garden center today. They're extremely large plants, very fragrant. Most people don't think of fragrance when they think of a begonia, but all of those traits are still there. And what plant breeders are doing today, and what they're really trying to put fragrance back in flowers. So they're going back into those old land races, pulling that fragrance characteristic out and putting it back into begonias so that you'll have a fragrant begonia and not just a pretty begonia. So knowing where these land races are and knowing where these wild populations exist provides an opportunity to further develop those products to regain characteristics that might have been lost over time. So we talked a little bit about the Columbian Exchange. This is when the old and new worlds kind of clashed into one another. A lot of people know tomato sauces as being, a big part of Italian cooking, but they weren't originally in Italy. They came from China. So when Marco Polo did his trips, he brought back those tomato varieties, and they were originally thought to be poisonous. So these cultivars have traveled all over the world multiple times. And it's very interesting to just kind of learn the history of those and kind of see where they came from. And it does provide a very deep pool for genetics for plant breeders to do their work with. So you can't talk about genetics without talking about our good friend Friar Mendel and his 30,000 peas. He was one of the early fathers of modern genetics, and he really discovered three key laws that are currently used today in all genetics, the basic genetics and understanding of how genes move and how they work. The first is a law of segregation. For those who may not be familiar with the graph on the right-hand side, that's a Punnett square. And basically what happens is you get one gene from your mom, you get another gene from your dad, and then as that zygote forms, they split and can split into different formats. So when you're talking about that law of segregation, pairs of genes separate. So on the top, you're seeing the two Ys that are going to give you a dark green pea. And then you've got a heterozygote on the left-hand side that has a yellow Y for a yellow bean and a small y for a green bean. So to get that green bean, you need to have two of the green traits. And a heterozygote is going to have one yellow and one green to show the color format. So that's how those genes actually segregate in the seed population, so that you can get a combination of multiple different types. So when a plant breeder is looking at segregation, they might actually do a cross to try and get to that yellow bean. And there's a little video that's going to show this in a little bit more detail, but those genes segregate. So they have to grow that population out the next year to identify and to select what they're looking for. So same type of thing, if you're wanting a tall plant, you're going to select for that tall plant, but those genes are then going to segregate out. So you're going to, perhaps when you grow that out, that seed crop out the next season, you're going to see some short plants. You keep selecting that tall plant over time, you're going to remove more and more of the things that you don't want until you eventually have what you're looking for. The law of independent assortment is that individual traits separate independently. They're not dependent on one another. Early on, people used to think that certain traits were connected to other traits in ways that they're really not. So genes separate independently of one another. And then there's the law of dominance. Some traits are dominant over other traits. In humans, for an example, eye color, brown is dominant over blue. So if you have two blue-eyed parents, you're likely going to get a blue-eyed child, but two brown-eyed parents, unless they're heterozygotes, are not going to produce a blue-eyed child. It's not possible with the dominance. So different traits are definitely dominant over one another, and you need to really understand that when you're looking at trying to develop genetic varieties. And if you're really looking for that recessive trait, your work gets a little bit harder because those heterozygotes are never going to show you that trait. They're always going to show you the dominant trait. Let's see here. A lot of this work was done in the early, the late 1800s, and Friar Mendel's work really wasn't appreciated until well after he had passed. People thought he was kind of a crackpot and didn't really know what he was talking about. So his work really got appreciated in the early 1900s, and it's still in kids' school books today. So what his gift to genetics is well understood and really appreciated. So what are the key things that you really need to do and have in place to do good plant breeding? We talked about those land races and where they were developed. So you really need to know where you can find that genetic pool. You need a good sized genetic pool to be able to make the modifications that you're looking for. Social systems became in place where, after World War I, farms needed crops that were more productive, more disease resistant. They needed better yields out of the land to make sure that we're able to feed people. And then the basic knowledge of genetics really grew and expanded quite dramatically in the early 1900s. There's also something that you need to kind of keep in mind when you're thinking about plant breeding, is it's also somewhat geography specific. There's a kind of something that's kind of discussed in genetics quite often, is this G plus E. It's your genetic makeup plus your environment. If you've got, it's going to give you the phenotype you're looking for. So if you've got a person who's genetically programmed to be 6 foot tall, but they're really underfed, malnutritioned, and everything as a child, they're never going to reach that full genetic potential. And that's the same with plants. You can have a plant that is, really destined to grow really well and produce a ton of fruit and very high yielding and, be really strong and disease resistant. But if that plant's put in poor soil, not watered, not fed, it's not going to perform to its full genetic potential. So that's where we talk about the genetic potential plus the environment that it's living in. And that being said, a lot of research and a lot of work done is now going into kind of looking at those environments where plants can thrive a little bit better. Drought tolerance is one that's definitely a major focus. We know our environment's changing, and drought is going to become a major concern for a lot of the agronomic crops. So people are actually doing work where they're stressing the plants out, growing them in drought conditions to see which ones are going to survive better. But it's still an environmental impact. that needs to be addressed. So, so those are the key ingredients that need to be put in place for plant breeding to go. So what are the primary objectives most plant breeders are looking for? Increased yield is obviously a big one when you're looking at agronomic crops, not so much when you're looking at flower varieties. You know, if they produce a pretty flower, that's great. Improve quality in some way, shape, or form, whether that is the distance between internodes on flowers so that the flowers are clustered better and they're not big and leggy. So there's a lot of different factors where quality can come into play, whether it's, you know, a deeper, darker red, you know, taller, stronger plants that don't lodge. Lodging is falling over in the wind. You know, some of the salvia varieties, the really old varieties that are around, you get a windy day like we're having today, and those plants could be laying right on the ground because they just can't handle the wind. So plant breeders have done work to find varieties that are less susceptible to wind damage. Another one is disease resistance and pests. This is a big one that we really look for as well, because as with anything, you put a plant out there and there's going to be a bug that wants to eat it, and there's going to be a disease that wants to kill it. So if we can develop plants that are disease resistant, it's going to make them stronger and be able to resist that disease, and also helps us prevent from spraying chemicals and things that we may not want to be spraying in our gardens to kill those diseases if they do come in. So this is a major focus for a lot of plant breeders as well. We talked a little bit about varieties that are well suited to particular soils and climates. Whether that be dry, colder, hotter, whatever it might be. These really stretch the plants from where they're normally found and how they can be used. But this is definitely a major focus with the climates changing as they are today. Varieties that resist to a plant habit issue, we talked about lodging a little bit, can also, you know, plant varieties that grow better in the sun. Begonias are known to be a plant that just thrives in shade, but there are begonias now that they've developed that grow in the sun. You know, as with most of my clients that come in, they want, you know, if they've got full shade, they want all the sun plants are going to grow, and the guys that have full sun want all the shade plants. So this provides an opportunity for everybody to have beautiful begonias in their garden. So that work's been done over the past probably 4 or 5 years. There's been some really great new begonia varieties that do really well in full sun. A little hard to convince some people of that sometimes, but they do. And then there's the rare occasion when you get something brand new and very novel, and a brand new species. Pet koas are that. About 7 years ago, these came out now, and it's literally a cross between a petunia and a calabrocchia. And what happens typically in those situations is you get something called hybrid vigor. So it takes the best of each of those two parents and those two crops and brings that into that new plant. So this is a beautiful series of plants that bloom very much like a petunia, but the foliage and the texture of them acts much more like the calabrocchia. So the foliage isn't sticky. They're much more tolerant to cool and hot growing conditions. They don't have that. If you water petunias, a lot of times they'll look awful for 3 or 4 days before they really perk back up. These pet koas don't do that. The blooms don't produce seeds, so they continue to bloom all season long. Sometimes the older varieties of petunias, once they kind of set enough flowers, they're kind of done. They don't really continue to grow very well. And they have a beautiful, strong, vigorous root system. They get that from the calabrocchia side. Sometimes your petunias cannot. So this is another example of how you can take two crops that are vastly different and breed them together to get something new. On the vegetable side, I worked with a company when I was at Johnny's years ago, and they created what they call kalettes. It's actually, it looks like a Brussels sprout plant. Instead of the round little cabbages that you would get like the Brussels sprouts, they're little, looks like little bunches of kale. So it's a beautiful new variety that was developed in the vegetable world, that, again, a cross between kale and Brussels sprouts, and that's what they got. So this is kind of where for kind of nerds like I am, where you get really excited. So you can kind of look at the new varieties and enjoy them, but also get excited about the science behind them. So how does this all work? The first and easiest way to do plant breeding is selection. And this is kind of just as the name says, it's based on going out, observing a trait, and then saving seed from the one that might be the tallest, the boldest, the reddest, whatever you're looking for, and then replanting that next year and continue just to select for that single trait that you're looking for. This has been done for years. This is how plant breeding got started, and this is something you can easily do right in your own backyard. I was speaking with one of our clients, and she's found some cosmos in her garden that she absolutely loves. So I've been working with her, and over the past 3 or 4 years, she's been, collecting the seed from that cosmos and replanting it out and saving seeds from the varieties that she really likes. And this is kind of a fun little project for her, but it's something she can do right in the backyard to kind of create her own variety of cosmos that she really likes, and it's working well for her and her specific environment and yard. Traditional plant breeding gets a little bit more complicated. This is where you're actually controlling the mating of the plants. I'm going to talk a little bit about a plant anatomy here. Is that most plants have both male and female parts within them. So this is where a plant breeding, and we've got a video on this to show you specifically how this is done because it's kind of cool. They actually take pollen from a male plant and actually use that pollen to rub upon the female plant to create a variety that they're very specific looking for. So a male plant might have, or the plant that they're looking for that they're going to use as the male might have a very specific trait that they're wanting to get into that population. So they're going to use a female plant that has a similar trait that they're looking for, or another trait that they're looking for, so they can actually stack traits up this way. And that way they actually control the very specific breeding of that plant. They're not waiting for Mother Nature to kind of create that cross as you would with just basic plant selection. And, in basic plant selection, you're just kind of collecting it. Mother Nature's kind of created that, where bees have cross-pollinated whatever, and you're just hoping that you get the cross you're looking for. This is more controlled. You're actually able to take that male and that female, create the offspring that you're looking for, and hopefully that has the desired trait that you're looking for. So this is also known as traditional plant breeding, and this has been done since probably the early 1900s. And this is where Gregor Mendel kind of talked about those traits and controlling those traits, and this is how traditional plant breeding kind of came about. Back in probably the late 80s, early 90s, molecular assisted plant breeding came on the scene. This is not genetically modified, but it's using and understanding the genetic code for different traits and actually screening the plants prior to even planting them in the field. So you can actually take a little, it's literally looks like a hole punch off a piece of a leaf, test that to see if it has the trait that you're looking for before you even plant it. So it's a much quicker way to understand whether or not that specific plant has the genetic makeup that you're looking for. But what that requires is that you understand the gene involved and you understand what sequence it has so that you can go look for it. But this considerably speeds up plant breeding. So you're not actually putting a bunch of plants in the field, waiting to go look at them before you're actually planting them out. So molecular assisted plant breeding does dovetail very nicely with traditional plant breeding. And it's used quite widely right now, especially with the agronomic crops. And a lot of flower breeders are using it as well now, especially when it comes to disease-resistant traits that they're looking for. The one everybody hears about a lot of times, and as soon as you say molecular anything, they assume that it's genetically modified. Genetic modification is where you're actually taking a gene and DNA from a completely different species and putting it into a plant. There's no way this could naturally occur. You're not going to be able to produce this in nature. You're not going to hope that a gene from a frog jumps into a plant. It's just not going to happen. But what they're looking for in creating this is they know that that specific gene provides some sort of trait characteristics they're looking for. And it's generally a resistance to sprays and things like that, so that they can go in and they can spray a field kill the weeds without killing the plant. That's a lot of times what they're looking for with genetically modified plants. Or some color enhancers and flavor enhancers that I don't know if I really buy that they're much better, but they do it. The last one, and this is a new one that's kind of come on the scene probably in the last 10 years. It's a gene editing technology called CRISPR. This is not genetically modified in the way that you're taking a gene from one species and putting it into another. It's actually like editing. You can literally go in and replace or change one or two DNA sequences, base pairs in the gene that you're interested in changing to get a different characteristic that you're looking for. This has also been widely used in medicine today. It's starting to get used more and more. And it's a fabulous technology that the Dudna and what's her, Sharpella, I think is her last name, from France. They were given the Nobel Peace Prize for developing the technology. So if you haven't heard about CRISPR yet, you're probably going to hear more about it for definitely some new technologies for how to treat diseases. So those are kind of the methods that are used. This is a quick little video to kind of hopefully explain a little bit better than maybe I did on what this is all about. So hopefully this will work. Hopefully that gave you a little bit. All righty, I went out of here now. There we go. Oops. Hopefully that gives you a little bit of a better kind of, I know it's cartoony, but a little bit better understanding of how those crosses work. And it can take up to 10 years to get from that first cross that you've made until you have what you're looking for. And that is actually even growing that out multiple times in different locations to get to that trait that you're looking for, that blue tomato. Oftentimes plant breeders will breed in the summer here, and then they'll go in the summer southern hemisphere because it's summer there in the wintertime here. So they continue that process. They don't have to, they're not missing a season, but it can still take quite a while to get where you're looking for. So I'm amazed by genetics and by plants and the power of selection that can occur. Brassicas are a very large plant group, and just kind of looking at what you can do and what plant breeders have done over time is kind of amazing. From a common wild mustard, a brassica that was found, over time, all of these different crops have been selected from that basic genetic makeup from that wild mustard that they found in nature. So if they were selecting for better root development, you get turnips. For better flower buds, you can get cauliflower. For leaves, you can get kale. And then you can continue on and cross like the kale with the Brussels sprouts and actually get something brand new and different like the kalettes that we talked about a couple minutes ago. So the power in what you can do is very amazing. Again, it takes somebody that's got a creative mind and kind of looking for different traits, and then the time and effort and dedication to actually get there. So this is kind of the basics. We've talked, we mentioned this a little bit earlier, to actually do the plant breeding and selection. Most of the, most plants have male and female parts. The male parts of the flower are on the right side. They include the anther and the filament, and that's where the pollen is created on that top of that anther. So when you're looking at a plant and you're pulling it open, you're seeing all that beautiful yellow pollen that's making us sneeze in the springtime. That's where it's coming from, and that's the male part to the flower. The female part to the flower is in the center there. That's the stigma and the style, and the embryo is at the bottom. So that's kind of where the baby plant is. And if you take the pollen from that male, it's going to, in this case, it's going to, if you keep everything together, that male is going to provide pollen to the female to create the plant. But in plant breeding, what they do is they would actually snip off the male parts to the flower. They call that emasculating the female flower. And then we'll take the pollen and the anthers from a different male plant with a trait and characteristics they're looking for, sprinkle it on top of that stigma. They close up that flower so nothing else can pollinate it. And then they tag it, follow it, let it grow out, and save seeds from that specific fruit or flower that they've created. So it's quite a tedious process. Some plants have very large male and female flower parts. When you're thinking about like a cucurbit, like a pumpkin, you can actually go out and you can see the embryos at the end of some of those flowers. The male flowers just have a stem coming through them. Those females have that really round ball at the end of those flowers. So cucurbits are pretty easy. When you get into tomatoes, I mean, you need to have some pretty good eyesight and some pretty tiny tweezers to actually be able to get in there and pull apart those flowers and to do the breeding itself. Okay, I think we kind of talked about all this. One more quickie little video here. Oops. Why? Come on. Oh my God. Let's see if this does it. Well, I can go find the video if you give me just a second. I'm sorry. Oops. What did I do? There we go. I was working on QuickBooks earlier today. How to move this. There we go. Tested this earlier, so I'm not sure what happened.
When did you find it?
Yeah, you're not seeing it?
No, we see your screen that says wrap it up.
Huh. Let's see. Let me see if this will share it. It's on my screen. Your screen is paused.
Are you sharing? There, we see something now.
Okay, perfect. Yep, I had to reshare.
Okay.
Let me start this from the beginning. There we go. Sorry about that. You got it?
Yes, but no volume.
Oh. Well, we can just watch it. She's talking
Okay.
about characteristics that probably over it, but she can show you, she's going to show you the process.
Okay.
So this is where she's getting in there and she's going to start removing the male parts of that pea plant. So she's actually starting to pull those filaments and anthers out to emasculate that female. So she's pulled out all the male parts to that female flower. Now she's found a male plant that she has a trait that she's looking for. But she's going to pull the pollen from that male. And then she's going to just dab that on the top of so she's labeling it to indicate exactly what cross she's made, so that when that variety grows out and those seeds are developed, she knows that it came from one male and one female plant. And she's closing that down so that it doesn't cross. So he's just going to talk for a couple minutes about basic plant breeding, but since you can't hear it, we won't go there. Okay. So screen share, here we go. Come on. All right. Come on. There we go. So this is the last slide. So just to wrap it up, you know, agriculture started out with the question of, did we kind of settle for beer or bread? And I think most people have decided that it was beer. And that really started the whole plant selection process, and really settled people down into towns and villages and cities eventually. So the real focus of plant breeding is to modify a plant to have whatever desired trait you're looking for. Whether that be, you know, a physical characteristic like height, a disease-resistant characteristic, color, but the goal is to modify that plant in some way for a trait that you're looking for. So breeding methods can go from very complex, from very basic to very complex. Again, stuff you can do right in your own backyard. If you have a pair of tweezers, and you can do exactly what the lady was showing you to emasculate a female, pollinate it to get the varieties you're looking for, or just do basic selection. I like this variety. I'm going to keep the seeds from this one and keep growing it out year over year until it really develops that full trait that I'm looking for. Understand that this is not a quick process. It does take a lot of time. It can take 10 plus years to get from the beginning of an idea that you have about a plant until you actually have it in hand and have it ready to go to market for clients and customers to use, and for you to see it in the catalog where it says new, you know, red begonia or scented begonia. You know that that has taken, and hopefully you can appreciate how long it's taken to get from a non-fragrant begonia to a fragrant begonia. So that's kind of what I had prepared. I'd be happy to answer any questions. I know I kind of went through some stuff fast, and hopefully I didn't confuse anybody.
Thank you, Wendy. I don't think you confused, well, you didn't confuse me. It was so interesting. I love that you gave us the backstory too. That was really cool. Ernie and Cindy had asked a question and it seems you answered it, but I'm just going to read it to you. Can plant makers move outside the genus to make these crosses? I'm thinking about all the different forms of, how did you pronounce it?
Brassicas?
And prunus. Sorry, there's no way to italicize or underline the Latin. And there are all of these new hybrids sterile, ? And then they then said, you've got ESP.
Yeah, well, hybrids are not sterile. And that's something a lot of people kind of question. And we'll talk about the ethics of plant sterilization in just a moment. But if you have a hybrid plant that has been created from a male and a female, and you save seed from that plant and plant it back out, just as they showed in the video, it is going to segregate back into a population. You're not going to get that hybrid again. Because that hybrid was generated specifically from that male and that female. And because of the laws of segregation, that seed is then going to segregate out into its parents again. So hybrids are not necessarily sterile, but you're not going to get what you're looking for. You're not going to get that original variety that you used. Open pollinated plants that are not hybrids, they should be true to type year over year. So when you go and you look at the seed package, you look at seeds, a lot of times you'll see OP behind that name, and that means it's open pollinated. That means you can save seed from that plant, and it's going to be pretty much true to type next season. If it says hybrid or F1, that is going to segregate out on you. So if you save seed from that plant, it is not going to look like it did the year before. It's going to segregate into its two parents. And sometimes they could be vastly different looking.
Interesting.
Does that make sense? And then in terms of seed sterilization, there was a lot of discussion around companies that we won't name names, that they were trying to work on creating hybrids that were sterile and that could not even produce seeds its next generation. That got a lot of very, 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 very bad backlash, because if those traits actually got out into nature, which they likely would, it could be very disastrous to the landrace species and other species that are out there. So while it is possible to create sterile plants so that they can't reproduce into another generation, it's not been done in any large way. Did I answer the questions?
I think so. We've got a thumbs up. Maybe we could stop screen share and just,
okay,
if anybody wants to come off or come on video. And Wendy, can we get you back on video?
Yes, there we go.
There you are. Anybody have questions? Ernie and Cindy wrote Monsanto ? Wondering if, yeah, okay.
I was trying to be kind.
If anybody has a question, you're welcome to ask, come off mute or put it in the chat. It's a quiet group. Just wait a few more seconds just in case people are typing. But wow, what an overview, Wendy. This was really fascinating. For those of us who just like a pretty flower, you are absolutely right. There's an appreciation that needs to be there, right?
Yeah.
Pretty amazing.
Things are always more complicated than we ever think they are when we're kind of just looking at something.
Yeah. Yeah. Well, I don't see any more questions in the chat. And so unless people want to come off mute, I'm going to close out for tonight then. All right. Again, Wendy, thank you very much for joining us and for for giving our patrons this education. And we will have it on our website for others to view, for sure.
And if anybody ever has future questions, just stop down to the garden center. I'd love to talk plants all the time.
Yes. All right. So this concludes our presentation for this evening, and all of us at Vose Library, thank you for attending our Vose Virtual Wednesday series, and we hope you'll spread the word and join us on Wednesday, March 2nd, when Earl Smith, author of Maine Inventors, discusses his fascinating book. So thank you, Wendy. Thank you, everybody. Good night.
Thank you, everybody.
Bye-bye now.